Okay, next is the secondary constriction or the nucleolar organizer. So the centromere was the primary constriction, no? If you remember, I told you that the centromere is not a bulb, it is a constriction, no? It's like a squeezed part of the thread which will attach the spindle fiber, no? Okay, so other than the main centromere, okay, other than that, there are other constrictions. Okay, let's say this is your centromere in between, no, here, and then the spindle fiber will attach from here. Okay, then maybe in another part you have another constriction like this, and then the rest of your chromosome, and then from the other side. Okay, so this could be your secondary constriction. Okay, so let's see what they've written there. How do I zoom? Okay, that's better, I think. So the chromosome, besides having primary constriction or centromere, it also possesses secondary constriction at any point of the chromosome. Okay, at any point. They are constant in their position and they are useful in identifying particular chromosomes in a set. Okay, so a particular chromosome will have a secondary constriction at a particular place. Let's say this is chromosome maybe 3. Okay, and let's say this is chromosome 4. Maybe it has somewhere there. Okay, the other arm. Okay, so something like that. So by looking at the scientists will say, oh, this constriction is over there in this arm, in the P arm, and this constriction is there in the Q arm. At that place, it must be chromosome 3 and 4. So to distinguish between the primary and secondary constriction is because the chromosome, they bend only at the primary constriction or the centromere during anaphase. Okay, so it will the spindle fibers, they know, okay, they will never ever bend or attach to the secondary constriction. Okay, even let's say by mistake, it will not bend there, it will bend from here when the chromosomes get pulled apart. Okay, it will bend only at your primary constriction. The chromosome region that is distal to the secondary constriction, that is region between secondary constriction and the nearest telomere, is known as the satellite. Okay, so let me draw something like that. Let's say this is your primary. This is the primary constriction, and let's say this is the secondary constriction. Secondary. Okay, then the ends of the chromosome, this end and this end, like the tip of your fingers, no, this is your telomere. This one is also your telomere. Okay, so this end and this end of the chromosome, like your fingertips, no, those are the tips. So the tips of chromosomes are telomere. So the region that is from the telomere and a secondary constriction, that is known as your satellite. Okay, the region between a telomere and a secondary constriction, this region is known as your satellite. Oh yeah, this is the word, this word, satellite. Okay. So therefore, those chromosomes that have secondary constriction are called as satellite chromosomes or SAT chromosomes. Okay. So the number of satellite chromosomes or SAT chromosomes, they are different in different species. The nucleolus is always associated with the secondary constriction of the satellite chromosomes. Therefore, secondary constriction are also called as nucleolar organizer region. Okay, let me draw you a cell. Okay, this is your cell. Okay, cell wall, cell membrane, no, then cytoplasm, and then your your cell organelles, your ribosome, your mitochondria, etc., etc. Then you have your nuclear envelope or nuclear membrane. Then you have the nuclear material. Okay. Then you have your nucleus. Okay, this is your nucleus. Then you have your, see, this whole thing is your nucleus. Then you have your chromosomes, no? Okay. Then inside the nucleus, there is a small round and dark region. Okay. This small round dark region is known as your nucleolus. Okay. Let me repeat. Inside the nucleus, okay, cell wall, cell membrane, etc., etc., cytoplasm. Then this is the nucleus. Then here are your chromosomes. No? Then inside the nucleus is your nucleolus. Okay, it's a darkly stained region. Okay, it's round in shape. So that is your nucleus. Sorry, not nucleus, nucleolus. Okay, so this nucleolus is always associated with the secondary constriction of the satellite chromosome. And it does the work of organizing the nucleolus. So the secondary constriction are known as the nucleolus organizer region, and satellite chromosomes are often referred to as nucleolus organizer chromosomes. Okay. Nucleolus organizer region of each satellite chromosome contains several hundred copies of the gene coding for the ribosomal RNA. Okay. So this nucleolar organizer region means the satellite part. No, it has the genes that code for your rRNA. Okay, this is your net or your entrance question. Okay, fine. Let's go to the next. Now looking at the telomeres. As I told you, these are the tips of the chromosome. Okay, so let's see how they are written. These are the specialized ends of a chromosome which exhibit physiological differentiation and polarity. Okay, it means like they have charge. No, they have they are different. They are different from the rest of the chromosome. Okay, this is your primary constriction. These telomeres, so these ends, they are different from the rest. Okay, from the rest of your chromosome. They have different physiological makeup. They are polar. Okay, it means they have direction. And then okay, and they are at the extremes. Okay, at each extremity of the chromosome, due to its polarity, it prevents other chromosome segments to be fused with it. Okay, so why does it have polarity? Is so that it will, even if you cut, okay, let's say some injury happens and you cut this chromosome. Okay, you use x-rays and you cut. If you cut it away from the telomere region, what happened is this. Okay, this chromosome, by mistake, it can mutate and it can join with another maybe cut chromosome. Okay, this side has telomere, this side has telomere. So it can cut, it can lead to mutation. So what nature did, or what God does, so depending on what you believe, we have telomeres, and these telomeres, they have the polarity, which signifies that this is the end of this chromosome, nothing else to attach. Okay, and this polarity, the charge, it repels. Okay, like magnet, no? The charge, they repel, right? They repel or attract. So depending on it will the charge of the polarity of the telomere, they will push away the other chromosome and it will not allow the other ones to stick. Okay, so I think it's clear enough. If any doubt, you can always ask me, you can always text. Okay, let's continue. So the ends of a chromosome are known as the telomeres. Okay, this line. Okay, if a chromosome breaks, the broken ends can fuse with each other due to the lack of telomere. Okay, I've already explained you. Okay, before reading this, I already explained. If you cut a part without a telomere, it can fuse. Okay, it can fuse. This is how mutations happen, no? Okay. Okay, now looking at a karyotype and idiogram. So we all have a set of chromosome. I have 23 pairs. Okay, we humans, we have 46 total number of chromosomes, and then I have 23 pairs. Okay, 23 pairs. We all have 23 pairs. Now, if I arrange all the pairs, this is pair 1, pair 2, 3, 4, 5, 6, 7, 8, 9, so on and so forth until your 23rd. So I have arranged all of them, okay, and I have arranged them by size, okay, by shape. All right, that arrangement, okay, that in simple words, that arrangement is known as your karyotype. Okay, did I put an image? Oh yes, something like this. 1 minute. Okay, see, this is a karyotype. Yeah, this is a karyotype of a human being. Okay, of me, of you, a human being. So we have, not we, means the scientist has arranged these chromosome pairs, these 23 chromosome pairs according to size, right? From the biggest to the smallest. Okay, the smallest will be obviously very small is the Y chromosome, no? And the pair of Y chromosome is the X chromosome, no? So he pairs them according to size. So from the largest to the smallest, okay? So there, chromosome 1 is arranged with its pair, 2 is arranged with a pair, 3 also with its own pair, 4 and 5, and so on and so forth. He has arranged them according to their size, their shape, their structure, no? Where how they pair means he must have observed them properly during meiosis or mitosis, and then he could arrange. So this arrangement, no, is known as your karyotype or karyogram. Okay, so let's see what they have written. A group of plants and animals, maybe I should make it bigger so that you can read. Okay, a group of plants and animals, they comprise comprising a species characterized by a set of chromosomes which have certain constant features such as chromosome number, size, shape of individual chromosomes, etc. Okay, so the term karyotype has been given to the group of characteristics that identifies a particular set of chromosomes. A diagrammatic representation of a karyotype as a species is known as idiogram. Generally, an idiogram, the chromosome of a haploid set of an organism are arranged in the series of a decreasing size. Okay, so an idiogram, no? See here. A karyotype, right? This person arranged them pairwise, okay? But in an idiogram, the scientist is arranging them not as pairwise but as size-wise, okay? Because even though they are paired, if you look, let's just go up again. If you look up there, if you have a look properly at the chromosome, even though they are the same pair, there's a little bit of a change in size. Do you notice this? Then this one also, plus minus some difference of size is there. Here also, this chromosome 20, there's a difference in size. Okay, let's. So even this chromosomes 3, do you notice? They do have a sort of a difference in length, okay, even though they are homologous pairs. So an idiogram, the scientists, they arrange them according to their size, okay, because even though they are paired, the sizes, they are somewhat different, okay. So here this ideogram, they are arranging in decreasing order of size. But in a karyotype, they are arranging them according to pair, okay, from the largest pair to the smallest pair. I hope the basic idea is clear. I hope the basic idea is clear, girls. So let us look at what they say. Obviously, I will read, you know, better always to read what the author has written. But I hope that at least the basic, you know, you have a basic idea. So let's go through. A karyotype is a diagram that shows the chromosome number and constitution in the cell nucleus. It consists of a whole set of homologous chromosome pairs. Okay, so like I said, set of homologous chromosome pairs arranged in decreasing series of their size. Okay, so arranged in decreasing order of their size. Microphotographs of metaphase chromosome help in lining up the homologous pairs and the preparation of a karyotype. So they are taking microphotographs, okay, during the metaphase time. So the karyotype, it reveals information regarding the number, the size, the shape, the position of the centromeres of each chromosome, the length of the chromosome arms, the place of the secondary condition, the satellites, etc. Okay, so karyotype gives us all these information. So by analyzing the karyotype, how much? Okay. So by analyzing the karyotype of the organism, we can detect genetic disorders and other information. Let's close this so that there. By arranging the 1 minute, let me just pause. Okay, so why do we need to study the karyotype? Okay, we study them so that we can detect genetic disorders. Okay, like Crohn's disease, like so many diseases. No, I forgot. What's wrong with my brain today? It's not working. So many genetic diseases, no? And other information about the individual, okay? It also tells us the gender of the individual. Well, obviously, whether you have an X or a Y chromosome. It can also tell the species to which the individual belongs, okay? Suppose blankly, I just collect some samples and it's all got mixed up. So, you will tell, Miss, I'll ask you how to identify. You'll say, Miss, let me prepare the slide. And then I'll observe these, I'll take the micro photographs and I will observe the size, and just by the number and the pair of chromosome and their sizes, I will tell you which species. Okay. So the karyotype can determine whether the individual has a chromosome disorder which leads to genetic diseases such as Down syndrome, Klinefelter syndrome and other ploidy conditions. Okay. Then let's just finish this ideogram also. I know it's almost 20 minutes beyond the listening capacity, but just a little bit more. Okay, so ideogram is a diagrammatic representation, okay, or it is a schematic diagram. Okay, so this is a diagrammatic representation. Okay, like your graph. No, if I ask you to define a graph, you will tell me it's a diagrammatic representation. Okay, in the form of pie chart or histogram. No. Ideogram shows the chromosome maps indicating the location of genes as bands. Okay, so it's a map that will show you the location of different genes as bands. Okay, so look here. So you can see some dark and light bands. These bands are all genes. Okay, let's say this is gene 1, gene 2, gene 3, gene 4, gene 5. Okay, so these bands, they show you the different genes. This chapter is really long. Okay. It is not an actual picture of a total chromosome of a cell. However, the idiogram provides much information about each chromosome, and it shows us the location of individual genes present on a chromosome. So the most important function is to show us the location of the genes. Okay, so it doesn't really tell us much about the diseases, right? But we do get information about the genes, okay, because they show the genes as the bands. They are useful also in identifying various abnormalities associated with a range of chromosomal disorders and determining the link between the structural abnormalities and individual genes correlated with various diseases or syndromes. Okay, so karyotype, it will be for the Down syndrome, no, the chromosome 21, instead of a pair, it is in a triploid condition. Okay, the rest of your chromosome 1, then 2, then 3, so on and so forth, they're all in normal pair, but except chromosome 21. So that there, karyotype will help you, because just looking at the karyotype, you'll say, oh my god, chromosome 21 has 3 pairs, so this person will have Down syndrome. Okay, but in an idiogram, other diseases which are caused by genes, not by chromosome pair, it has nothing to do with whether one pair is missing or one pair is extra, we don't care about that, but there are some genetic disorders, no, like that wolf man disorder, no, they have very hairy face and all, no, so those were the genes that we had accumulated, no, those junk DNA as man evolved, no, maybe from hominoid species to australopithecus and all, no, so from tetrapedal we walked to bipedal man, no, from the hominids we came to Homo sapien. So maybe those genes got turned on by mistake, because our DNA, it is the biggest data bank. Okay, we never throw any information, we compact it, we save it just in case we may need to re-evolve again in the future. So those chromosome those are not chromosome disorders, but those are genetic disorders. So the idiogram will help us to study the relationship between these diseases. Then I think similarity and difference and the uses, maybe we should go to the next class as it's beyond 20 minutes. So I'll see you in the next class, girls.